From Diamond Light Source, this is the Diamond Podcast.
This month, we speed up your lives with the power of catalysis to discover how elements and compounds can be used to improve the production of biofuels, give a kickstart to industrial reactions, and remove harmful pollutants from our atmosphere.
The material has very excellent properties in the selective capture of CO2 and SO2, while all the other common small molecule gases, such as the hydrogen, oxygen, nitrogen, carbon monoxide, can pass through the material with very little uptake. Actually, the uptake would be 100 times lower than that of CO2 and SO2.
Sihar Yang will be explaining how metal organic frameworks are being used to accomplish this later in the programme, when we also visit the new Catalysis Hub on site at the synchrotron, as well as hear the latest news from Diamond, including the potential for a male contraceptive pill. So all that to look forward to in this June edition of the Diamond Light Source podcast with me, Mira Senthilingam.
The Diamond Podcast. For more information, look us up online at diamond.ac.uk/podcast.
Catalysts are used throughout industry to increase the rate of a chemical reaction. Their presence helps a reaction take place without the catalyst itself being used up in the process. As a result, they're crucial to many industrial processes, including the Haber-Bosch process used to produce ammonia, as well as the more domestic catalytic converters found in car exhausts. Scientists across the globe are constantly working on improving various reactions, and now a hub for such scientists has been built in the UK, on site at the Diamond Synchrotron, a catalysis hub, as Peter Wells explains.
So the UK Catalysis Hub is a new initiative that was funded last year by the EPSRC. It acts to bring together all the major players in catalysis in the UK under one organisation and seeks to advance, promote and coordinate UK catalysis research efforts.
So a wide range of catalysis research taking place here. What do you particularly focus on?
My main research interest is in studying catalysts under realistic conditions using the light source at Diamond.
And what types of catalysts do you focus in on?
So my background's mostly on electrocatalysis or heterogeneous catalysis. So this is catalysis for fuel cells or gas phase catalysis processes.
And what do you really need? What do you look into there? What are you trying to say improve? I guess improving the reactions themselves.
So we're trying to see how these catalysts work under realistic conditions, seeing what's absorbed on the surface, how the catalyst structure changes, and then trying to relate that to the performance so we can fine-tune our catalyst to make more active, efficient and environmentally friendly materials.
And do you have an example, say, of a particular catalyst that you've been looking at in real time, in situ, and what kind of changes or effects you were seeing?
One of the recent pieces of work we've looked at is a methanol oxidation catalyst. This is an imolibdate system, and the phases present in that material are very important for the catalytic activity. The activation step for these materials has a calcination process at higher temperatures, and we're able to study this calcination at these different temperatures and work out exactly what conditions we need to make the active phase.
So really you can then identify the conditions you need to have the catalyst performing at its best phase?
Exactly.
And where is this, say, catalyst useful?
In methanol oxidation to formaldehyde. It's a very important product industrially.
So we're currently in the catalysis lab here within the catalysis hub, and this is a very new lab. In fact, you've got kit arriving all of the time. So tell me a bit about all the different equipment we have around us.
So in this room here, there is some items of kit for catalysis testing, some for catalysis characterization, and also some for preparation.
So behind me, there's a big glass cabinet filled with 3 metal pieces of equipment with lots of kind of gauges on them.
So these are 3 high pressure reactors that we would use to study how catalysts operate under high temperature and high pressure.
So you would essentially just set different pressures and identify the best pressures for particular catalysts?
Yes, so this is a batch reactor. So we load each reactor with the catalyst, the substrate, and also we can pressurize with different gases. And then we can, at the end of that process, we can look at the products that are formed and quantify how good the catalyst is at working.
And over to the right, there's something possibly maybe more relevant to your work. So it's something that replicates the kind of in situ analysis that's possible at Diamond, but on a smaller scale here. So it's a kind of white box that's about 0.5 metre or so in height and about 1/4 metre in width. I mean, it doesn't look particularly big compared to, when you go and look at the size of a diamond. What goes on in here for you to see things in situ?
So Diamond have developed a quartz capillary microreactor that we use for studying gas phase catalysis in situ. And what we have here essentially is a commercial variant of that. We have a quartz capillary, a furnace that goes in, and we have a mass spectrometer for measuring the gases. And this allows us to sort of, do the same type of system, type of experiment here as we would do for those in situ studies.
And so, I mean, are they to the same level, or how do they differ at all?
When you do something in situ, you're compromising your reactor design to be able to perform that measurement. So the results that we get here are probably much more useful catalytically, but results we get at Diamond are much more useful for understanding how the catalyst actually works. So being able to marry those two bits of data together are actually quite useful.
So how does this kit all come together? There's a computer attached, there's the mass spectrometer attached, as you mentioned. So could you give me an example of a particular experiment you would do here?
So going back to that methanol oxidation catalyst, what we would tend to do is we would load the quartz capillary with our imolibdate catalyst. This is the active catalyst we use. And then we would, in a stream of helium, dose methanol over the surface. The methanol absorbs on the catalyst surface. Then we would heat under helium and look at the products evolved. And by analysing all the products evolved, we can see how selective the catalyst is for this reaction.
And so the mass spectrometer would show you...
This will give us the mass of the products given off, which we can then relate to the products in the reaction.
So really a lot going on here, looking at different aspects of catalysis. And, I mean, the hub is quite new. So what's really, I guess, the goal in the future or over the next few months or years, I guess?
The ultimate aim is to have a national centre for catalysis on this site. And this is, I guess, the first steps towards that. And I think it's a whole new way of working for catalysis in the UK. And to show that all these university groups can work together, coordinate in the national interest is a good way of working.
Peter Wells, Research Fellow at the UK Catalysis Hub. Now, this hub aims to bring together scientists working in this field. Scientists such as Sihai Yang from the University of Nottingham, who's looking to develop catalysts in combination with metal organic frameworks to store gases such as carbon dioxide.
My research area focuses on the development of a new porous material for gas storage, separation, and capture applications. Traditional porous material include zeolite and activated carbon, which has been studied for many years. Over the past 10 years, a new type of porous material was discovered. It's called metal organic framework materials, or for short, people usually call them MOFs. MOFs are usually constructed from metal ions or clusters bridged by organic linkers to give a three-dimensional extended framework structure. The most important feature of this type of material is they have very high porosity and large surface area, which are the key parameters for the gas storage applications. The pores in these MOF materials would be empty and available to store gases, such as hydrogen, nitrogen, methane, CO2, and so on. So my current research aims to develop new MOF materials for selective capture of harmful flue gases, especially for carbon dioxide and sulfur dioxide, because of their significant environmental impact, for example, the formation of global warming and acid rains.
And so there are many different groups, I guess, working on many different types of metal organic frameworks and different structures, all, I guess, trying to capture as much carbon dioxide as possible. What's your particular take, then, on the design of a MOF? So
in particular, we have recently created a new MOF material that shows great advantage in the carbon capture and storage applications. The MOF is called NOT 300 and was made from aluminum salt and a cheap organic material in water without usage of any organic solvent. And typically, in the MOF synthesis, organic solvent is necessary for the formation of MOF material. While in this case, NOT 300 doesn't need any organic solvent, so it just needs pure water as the solvent to make the MOF. So this synthesis can be viewed as a green synthesis. And the material has a very excellent properties in the selective capture of CO2 and SO2, while all the other common small molecule gases, such as hydrogen, oxygen, nitrogen, carbon monoxide, can pass through the material with very little uptake. Actually, the uptake would be 100 times lower than that of CO2 and SO2. In this way, this material is able to selectively grab the CO2 and SO2 from a cocktail-like gas mixture.
How did you really come up with this design? So how do you, say, use the beamlines at Diamond, and what do you really investigate in order to see the best combination, I guess, of these different molecules in order to capture as much gas as possible?
So the synchrotron facility such as Diamond has played a very important role. Over the past 5 years, we majorly carried out 2 types of experiment in Diamond. One is a high-resolution diffraction experiment for structural determination of our MOF materials, and the second one is the in situ study of a molecular mechanism by which the gas molecules are trapped and separated by using these MOF materials. Well, learning the crystal structure for these MOF materials is very important to understand their excellent properties and functions. MOF materials often crystallize in very small size, a few tens of microns typically, and if lucky, you may get crystals in 100 microns. The structural determination for crystals in such small sizes is very challenging. As I mentioned earlier, the high level of the porosity of MOFs, which is the most important feature of this type of material, will add more difficulties to this structural determination process.
What would you say the kind of main benefits have been then? So you've managed to investigate this particular structure of metal organic framework quite closely using the beamline.
Yes.
And how much have you found out about it really? And what really are the main benefits, say, of this structure, therefore, over other designs, other than the fact that it doesn't need an organic solvent?
Because the aluminum and the simple organic material, they have very good solubility in water. The nucleation process is different from the traditional MOF synthesis. Also, in the previous MOF system designed for the carbon capture, they usually incorporate a functional group called the amine functional group. And unfortunately, this group is toxic to the environment. While in the NOT 300 system, because it's made in water, so there isn't any amine functionalized group within the MOF host. Well, the only functional group within the host is a hydroxy group, and that was from the water and is non-toxic. And by using Diamond, we successfully identified the site of this OH group within the MOF host, and we successfully see the interaction between the adsorbed CO2 and SO2 molecule with this OH group.
And so how would this all really come together in order to capture these kind of harmful gases from flue gas? So you've got this porous material that can capture them. What do you do now in order to actually, I guess, use them out there to capture the gases you want?
Well, that's a good question. Actually, it's a little bit beyond our research. We currently are seeking for the collaboration with the industry and the engineers to test the material function and the properties in the larger scale, possibly in the real-world scenario, to assess the material function and the properties.
And just to finish off, really, through all of this work, recently you won the Young Investigator Award at Diamond. What did it feel like, I guess, to get this, award and recognition of your work in this field?
I'm really encouraged by this award. And also I will continue my exciting research program at Diamond and ICIS and try to uncover new exciting results with better material properties.
Sihai Yang from the University of Nottingham.
Did you know that Diamond's floor-level monitor system shows Diamond tilting twice a day due to the moon passing overhead?
This is the Diamond Light Source podcast, and this month we're accelerating through the science of catalysis. Still to come, we hear how catalysts are being used to improve the production of biofuels from everyday industrial wastes such as the byproducts of fermentation. But first, some news from the synchrotron, starting with some new insight into broken bones, courtesy of Diamond's communications officer, Mary Cruz.
Well, like many things in life, bones aren't perfect. Sometimes, if you're clumsy, prone to a scuffle, or a keen base jumper, bones break. But it's important to build our understanding of how and when bones break. This knowledge can help us to develop treatments for damaged bones and counteract the effects of bone-related diseases with targeted therapies.
So what's known, I guess, about our bones so far and its kind of structure?
Well, the toughness of bone and its resistance to fractures is a key indicator of the bone's quality. Bones which are damaged or depleted in some way as a result of nutrition or disease will break more easily than good quality bones. However, bone plays by its own rules. It fractures at different points and in different ways to other similar materials. It's particularly difficult to measure the fracture resistance and thus bone quality of smaller bones.
And why is that?
Well, these very little bones generally have a different toughness quality and behaviour to the larger ones. But it's really hard to measure, so our knowledge of the fracturing point of little bones is limited. And this has important implications because generally we use small animals, rats and mice mostly, to study the effects of various things, injuries, diseases, treatments or genetic abnormalities on bone quality. But as mice and rat bones are so small, the impact can be difficult to measure.
And so what have people been looking into here at Diamond then?
Well, a team of scientists have used the imaging station on I-13 to develop a new method of observing and measuring the cracking process that occurs when small bones break. The team looked at the way in which the bone whitens around the epicenter of a crack. So if, like me, you've ever bent a ruler in half really hard, you will have noticed a whitening effect around the center of the ruler just before it cracked. Now, this whiteness is caused by light reflecting on minute microcracks in the material. These microcracks occur around the damaged zone as the strain increases. They're a toughening mechanism designed to relieve some of the pressure on the main crack.
And so what did they actually find by looking into this?
Well, the team found that the observable changes to the coloration of the bone, it's not just the result of a toughening mechanism. They also indicate deeper damage below the surface in small bones. The team also showed that whitening can be used to track this damage, and so it can provide a useful tool to measure bone toughness. So we now have a new method of measuring the quality of small bone samples, and that's something that's really important to biomedical research into the effects and treatment of disease. And theoretically, this methodology could be used to measure the quality of other similar materials. As such, the findings could have important industrial applications.
Now, moving away from our bones and to contraceptives, but specifically for men.
Yeah, that's right. This is a story all about the racy subject of contraception. The female contraceptive pill has a history that dates back over half a decade. The first FDA-approved pill came to market in 1957, and over 50 years later, about 100 million women worldwide now take the contraceptive pill.
But why, I guess, to date, hasn't this been possible for use in men?
Well, that's right, yes. The search for a male alternative has thus far proved fruitless, and the reason that the science behind the pill doesn't work for men is that a male pill would need to target many millions of sperm rather than just one egg. Scientists have also had problems because a lot of drugs may contain the right elements, but they're just not able to transfer from the blood to the testes, and so they fail to have a contraceptive effect. Currently, the only drugs in clinical trials are contraceptives that counteract the testosterone hormones.
So what have the scientists found?
Well, now an international collaboration of scientists have used Diamond's IO3 beamline to develop a potential male contraceptive pill that works by targeting the specific protein responsible for sperm cell production. The pill inhibits the aptly named sperm-generating protein bromodomain, whilst leaving the testosterone hormones untouched. And this could be a really significant step forward for male contraceptive science.
But how does it actually work?
Well, the pill uses a small molecule inhibitor with the catchy name of JQ1. And once ingested, what JQ1 does is it effectively moves from the blood to the testes and inhibits bromodomain. Mice treated with JQ1 were found to have a reduced sperm count, and their existing sperm were found to be less mobile and of lower quality. Despite this effect, their hormone levels remained normal. They were still regular dude mice, but the effects of JQ1 were also found to be completely reversible. When the mice stopped being treated, their sperm count and quality returned to normal. It's also important to note that mating behaviours weren't affected, suggesting that libido isn't reduced by the male pill. And there didn't appear to be any obvious effects in offspring produced during treatment or afterwards.
So how did this all really come together and what could it mean?
Well, if the studies are anything to go by, we may not be too far from having a male contraceptive pill akin to the female version, which can cross from the blood into the testes, impair sperm generation, and produce a completely reversible effect. More research still needs to be done into the long-term effects and the impact of JQ1 on different species. This research is still quite early on, but it's still a mammoth step forward in the quest to develop the elusive male contraceptive pill. If nothing else, we certainly now know what proteins to target to control fertility. So look out, bromodomain. Science is on to you.
Diamond's Communications Officer, Mary Cruz. Now, this month, we're exploring the potential of catalysis to speed up reactions that are currently crucial for future industry, but are proving a challenge. One field facing the challenge of high yields is that of biofuel production, and Edmund Sang from the University of Oxford is on the case to make this happen.
If you look at the biomass, basically is a carbohydrate, and in terms of the structure, they contain, basic unit. Everything is shared is the same unit, and they contain carbon, oxygen, and carbon-carbon bond. So what we have the interest is, can we be able to break the carbon-carbon bond and put the hydrogen back on, so then we can cleave these polymer unit into the smallest useful unit. In this case, is methanol. And the methanol, if we can produce it, there will be lots of advantage. So you can use it for fuel cell to power your all electrical devices. People consider to use it to power up their car.
What are the main challenges facing this area? So when it comes to producing methanol from biomass, what are the current kind of challenges and problems that mean that this isn't, say, our current main fuel?
Well, I think the key problem is there is no catalytic process to do this. The biomass is quite large in terms of the molecular size, and they don't have sufficient vapor pressure. And if you heat it to a high temperature, you do the catalysis at high temperature, you will generate lots of products. It's very expensive to separate those products in an industrial way. So the pleasant challenge is to selectively produce one product rather than many products.
I mean, I guess a main problem with this whole biofuel production area is that it really at the moment requires very high temperatures and very high pressures. So it's not very cost effective and it requires a lot of energy. So ideally with your catalyst, you'd be trying to kind of bring all of that down.
Precisely. So the way to control it is bring down the temperature and develop a specific active catalyst that can cleave the bonds or specific location of the bonds in the biomolecules, so you made one single product out of it.
And so how are you taking this on, really, with your catalyst? So what is your catalyst and how does it work to bring these conditions down and produce methanol?
Initially, the material we put in is small palladium metals, roughly about 1 nanometer, 1 times 10 to the minus 9 of a meter, sitting on the iron oxide, high surface area iron oxide. And after reduction and during the reaction, what happened is some of these iron will be reduced as from the iron 3+ going all the way down to iron 0, and then forming a bimetallic species with the palladium. So these, we believe, is an active phase, and from the XF evidence, it suggests the more the amount of palladium ion species coming out, the better is the activity. So what it does is when ethylene glycol molecules come over to this material, then they will form a very strong interaction and they absorb onto the surface of these species and they pull the bond and helping to cleave the carbon-carbon bond in the presence of hydrogen. So then we made 2 methanol out of the 1 ethylene glycol with hydrogen.
And so what have you managed to see with your catalyst to date? So has it been quite effective, and have you managed to see kind of good yields of methanol being produced?
Initially, the selectivity, that means it's going towards the methanol, is pretty low. It's roughly about 15 to 20%. But after when we put it on, then the palladium ion species come out during the catalysis, then the yield will, selectivity is sort of increased to nearly 80%. So it's pretty impressive selectivity. And that is a good starting point. At present, the conversion is still low. That means the yield is not yet reaching the industrial practice. We're busy in trying to improve the yield.
So now that you've managed to see this yield and you're trying to kind of improve it, what would ideally be the end goal with your application? So you've worked with, say, ethylene glycol as a starting point, which it's quite, I guess, a simple molecule as carbohydrates go. Would you be working, say, with more waste materials, such as, say, from paper, kind of the paper industry, or kind of wood chips and things like that, which I guess are much more complex, things like cellulose and lignin?
Yep. That's his idea, yeah. We use the ethylene glycol as a model compound so we can learn how to cleave the carbon-carbon bond without cleaving the carbon-oxygen bond. You can think about, going up to address those more useful biomass molecules like sugar molecules, like sucrose, glucose, fructose, and then palm oils, further up, and the cellulose, lignin, all these have a very closely related structure. And we're thinking about recycling bioproducts. For example, in paper puff industry, and there are lots of bio-waste coming out. And in fermentation process, there are lots of bio-waste. At present, they create a lot of environmental problem. So yes, we consider the new catalyst may be able to offer a new way to recycle those biomass or bio-waste.
Edmund Sang from the University of Oxford. Now, that's almost it for this month, but before we go, we hear back from Mary Cruz as she brings us some further stories from Diamond's Flash Fiction series, sourced from an anthology created from Diamond's light reading fiction competition, which ran last year.
The Beamline by Tanya Hirschman. We had to keep smiling because they spied on us as we queued. If someone let the corners of their mouth droop down, if they let slip even for a second, that was it. A Diamond light source uniform would swoop and they'd be gone. We all had different methods for making sure we appeared cheerful. Some just concentrated on the physical, so that the grin was more of an upward grimace, tiring on the jaw muscles. Others of us decided to think only happy thoughts. Let that beam come from within. Easier perhaps, but who had so many happy thoughts? Hours we waited, and really, if you had that many, why were you here? What did you want? We didn't know exactly what we were waiting for. We received the invites in the post. Everyone in town got one, but all they said was, find the secret to eternal joy, the time, the place, and the smiling rule. So we came and stood. The line did move bit by bit, and the front people did seem to enter the building, but no one came out that way, and so we had no idea what it was all about. There were rumours. A room shaped like a hexagon, but with more sides. Light like you'd never seen. There was a whisper of nirvana, of heights of ecstasy. We held on to our invitations, and we kept grinning as we inched forward slowly towards it, the destination where we would know it all, where we would finally and totally fly free from the misery of our lives. Those of us who kept our smiles by replaying happier times wondered if we were beginning to feel it already. Small hints, a tiny warmth, minuscule promises of what was to come.
Mary Cruz with a tale taking us through the Beamline, courtesy of author Tanya Hirschman. Now, that is it for this month, but join us again next time for more insight into the range of science taking place at the synchrotron, as well as more news and more fiction to get you thinking. In the meantime, if you have any questions about Diamond or the research taking place there, the email is podcast@diamond.ac.uk. You can listen to previous editions of this programme online at diamond.ac.uk/podcast or nakedscientists.com/diamond. And you can also subscribe to the podcast on iTunes. Thank you this month to Peter Wells, Sihai Yang, Mary Cruz and Edmund Sang. I'm Mira Senthilingam, and until next time, goodbye.
The Diamond Podcast is brought to you by Diamond Light Source and produced by thenakedscientists.com. There's more information on our website at diamond.ac.uk/podcast.